Yes.
Present.
Good evening, everyone. Yes, sir. This mean today my topic is brief mention on genus Pencillium. Yes. So Pencillium is a genus of saprophytic fungi. They are commonly known blue or green mold. They are economically important for the production of cheese, organic acids, and antibiotics. They play an important role as a decomposer in the ecosystem. Penicillium can be found at the various places such as soil, air, on decaying food, etc. Penicillin is one of the most important antibiotics extracted from Penicillium species. The wonder drug penicillin was first discovered by Sir Alexander Fleming in 1928. During his work with Staphylococcus aureus, get contaminated with mold spore, Penicillium notatum, which after proper growth caused death of Staphylococcus aureus, showing lytic zone around itself. He isolated and called this antimicrobial compound as penicillin. Later, it was found that a strain of penicillin Chrysogenum is most efficient than penicillin notatum in the production of penicillin. So when we see the classification of Penicillium, the domain falls under Eukaryota, kingdom fungi. Yes, sir.
Please move your slides.
Yes, sir.
Your slides are not moving.
So now, sir.
No. Shall I present from here?
Okay.
Huh?
Yes, sir. Yes, sir.
Okay, wait. Has been start presenting.
Okay. Sir. So next slide, sir.
Start from the first.
Okay. Sir. Yes, so genus Penicillium belongs to Ascomycetes group of fungi and is well known for its importance in antibiotic and food industries. So the name Penicillium is derived from the Latin word pencillus, meaning is nothing but brush-like structure, which refers a stalk-bearing radiating chains of conidia resembling like a broom. So Pencillium is a genus of saprophytic fungi. They are commonly known as blue or green mound. They are economically important. So previous slide, sir. They are economically important for the production of cheese, organic acids, and antibiotics. They play an important role as a decomposer in the ecosystem. Penicillium can be found at various places such as soil, air, on decaying food, etc. Penicillin is one of the most important antibiotics extracted from Penicillium species. The wonder drug Penicillium was first discovered by Sir Alexander Fleming in 1928. During his work with Staphylococcus aureus that contaminated with mold spore, that is Pencillium notatum, which after proper growth causes death of Staphylococcus aureus, showing lytic zone around itself. So lytic zone is nothing but breakdown of a cell. He isolated and called this antimicrobial compound as penicillin. Later, it was found that a strain of Pencillium, Chrysogenum, is most efficient than Penicillium aureus. So when we see the classification of fungi, so it falls in the domain Eukaryota, kingdom fungi, phylum Ascomycota, class Uroshiomycetes, order Uroshials, family Trichocomaceae, and genus Pencillium. So when we start to see the structure, so next slides. So it has a vegetative structure of Pencillium, that is, it is having a multicellular mycelium. So mycelium is generally made up of hyphae. So made up of highly branched, multinucleate, and septate long thread-like filamentous structure known as hyphae. The cell wall is made up of glucose, polysaccharide, and chitin. The cytoplasmic continuity is maintained through central pore present in the septa. So conidiophores are present at the branch ending along with the spherical conidiophores, which are asexual spores produced exogenously. Conidia are produced in the basipital succession, that is, the youngest conidium is present at the base. Conidia are produced from the specialized cells called phyllite present in the group and give a brush-like appearance to the fungus. Ascospores are found in asci arranged in ascus. So they are generally responsible for the sexual reproduction. Some of the mycelia grow deeper into the substratum to derive food. The food is stored in the form of oil globules. When we start to see the reproduction of the reproduction life cycle of Pencillium, so Pencillium reproduces with all three processes, that is vegetative, asexual, and the sexual. So vegetative reproduction is, when we see the vegetative reproduction, basically it is of three types. So that we'll be learning, like fragmentation, budding, and so on. So basically Pencillium will have a vegetative reproduction through a process called fragmentation, and each fragments develops separately, making a complete mycelium. Asexual reproduction takes place in the special structures called conidiophore. Asexual reproduction takes place by unicellular, uninucleate, non-motile spores, the conidia, formed on conidiophore. The conidia develops as an erect branch from any cell of vegetative mycelium. The branch of conidiophore is known as ramus, which further becomes branched known as medullae. A number of flask-shaped phyllite, or which is also called as sterigmata, develops at the tip of each medullae. Each sterigmata develops at its tips a number of conidia. During the development of conidium, the tip of stegma swells up and its nucleus divide mitotically into two nuclei, one migrates into the swollen tip, and by partition wall, the swollen region cuts off from the mother and forms the uninucleate conidium. The tip of stegma swells up again, and following the same procedure, second conidium is formed, which pushes the first one towards the outer side. This process repeats several times, and thus a chain of conidia is formed. The conidia are oval, elliptical, or globous in structure, having smooth, rough, and one of various colorations like green, yellow, blue, etc. After maturation, the conidia get detached from the mother and are dispersed by wind. On suitable substratum, they germinate by developing germ tube. So sexual reproduction is by the formation of ascospores. So some species of Pencillium are homothallic and some are heterothallic. So male and female sex organs will be present on, if they are present on the same thallus, it is no, and it is present on the different thallus, then it is heterothallic. So here the male sex organ will be antheridium and female sex organ will be ascogonium. So one of the cells of the vegetative mycelium develops into ascogonium, so which is unicellular, having a single nucleus. The nucleus of ascogonium undergo repeated divisions to produce 32 to 64 nuclei. The developing antheridium branch coils around the ascogonium, and the apical part of it is separated by septa, forming a unicellular antheridium having a single nucleus. After maturation, antheridium bends and touches the ascogonial wall with the help of enzyme. Here the cell wall will start to get dissolved, and both the cytoplasm of ascogonium and antheridium will start to get intermixed. This probably we call it as a plasmogamy. There occurs an intermediate dikaryon phase, which is a dikaryon stage with two nuclei, that is N plus N present in the cell. The ascogonium repeatedly divide with the partition of the wall, forming many binucleate cells arranged one above the other. The terminal dikaryotic cell swells up, forming ascus mother cell. So karyogamy is nothing but fusion of two nuclei occurs in cell-forming diploid cell. So basically it forms a zygote. The diploid zygote undergoes first meiosis and then mitosis to form 8 nuclei. Each accumulates some cytoplasm, resulting in the formation of 8 ascospores. The ascospores are released by the dissolution of ascus wall and get detached to a suitable substratum. The ascospores develop by forming a germ tube and mature into a branched mycelium. So here is an image of a sexual and sexual reproduction, where we can see how anthridium has been branching out in the ascogonium, and later on it is getting attached to the wall of anthridium. No, sorry, ascogonium. Next slide. Yes, so economic importance of Pencillium. Pencillium species are very important economically. They produce many food products, organic acids, antibiotics, and mycotoxins. So cheese production, many Pencillium species are used in the production of different kinds of cheese. Example, blue cheese, and so on. So enzymes and organic acid production. Many Pencillium species are used in the production of organic acids such as citric acid, gluconic acid, tartaric acid, and enzymes like amylases, protease, cellulase, lipase, and pectinase. Antibiotic production. In 1929, Alexander Fleming isolated the antibiotic, that is a penicillin. Apart from these, there are also certain uses of Pencillium. That is, Pencillium species are used in the production of antifungal drug and tumor suppressing compounds. Pencillium species are also used in mycoremediation, that is bioremediation process using fungus to clean the environment due to their ability to break down xenobiotic compounds. Thank you. Thank you, sir.